Well, thank you so much for joining us on this industry panel. It is very exciting to really show the amazing progress of people working on our disease. I'm Ben Lenail, and I'm an adrenomyeloneuropathy patient living in Palo Alto, California. I was diagnosed about 10 years ago, and I'm one of the co-founders of ALD Connect, along with some of the fine people we heard earlier today, such as Florian Eichler, Keith Van Haren, Josh Bonkowski. And what's exciting to me in this session is really to show our progress, because when we started ALD Connect in 2013, there was one company working on ALD, Bluebird Bio, and that was a primary goal of ALD Connect, was to drum up awareness and interest in this disease in the biotech community. And so today we have an industry advisory council that has 15 members. And on this panel today, we will hear from a few of the companies that are actively developing therapies for different types of ALD. What's interesting today is we have different modalities, different approaches. We have gene therapy, we have small molecules, we have stem cell treatments, we have some for pediatric phenotype as well as adult phenotype, and that could be relevant for both men and women, affected men and women. And we'll see that they're in different stages in the drug development life cycle. Some are early and some are closer to the bedside. But it is exciting to see the progress. It is inspiring. It is hopeful. And I hope this session will give rise to a lot of interest, a lot of questions around what patients can expect. So I will start by giving the baton to Joe at Bluebird Bio, since they were the company first to work on this condition. And so Joe will give us an update on Bluebird Bio's progress.
There we go. Thanks, Ben. Can you see my slide okay? Just confirm.
We can.
Great. So thanks so much, Ben, for the introduction and the background there. And also thanks to ALD Connect for inviting us to present a little bit about Bluebird Bio and who we are. As Ben said, we've been kind of working together for a long time now. And also thanks, I just recently came off the patient panel, and it's one of the reasons I love this meeting. I love what Florian and the team have built with this community. So thanks so much for sharing your stories. It keeps us, in the day-to-day working on these therapies, front of mind as to why we're here. So thanks so much for sharing that. I hope next year we can all be in person again, and I'll get to see you all, meet some new faces. So with that, I just have a few slides to share. So my name is Joe Biedenkapp. I'm a program lead for our cerebral adrenoleukodystrophy program. As been noted, we've been developing this program for some time. So just a few quick disclosures. This is an investigational therapy. It has not been approved by the FDA at this stage. And I am a full-time employee with equity in the company. And as a result, there are certain things I can and can't discuss today with you, but urge you to reach out to your physicians and many on this call who are clinical investigators for us with specific questions, but we'll certainly answer what I can. So just a few slides to run through, who Bluebird Bio is, set the stage and context for what it is we're doing with our gene therapy, talk at a high level about our clinical trials, one of which is still ongoing. And then, what's kind of fun to share is some recently launched patient and caregiver educational materials that we've partnered closely with ALD Connect and others in the community to develop. And they're all the better for it. So thank you for that. And we're really excited to share and have some of those things available to the community broadly. So with that, Bluebird Bio has been around over 20 years now. I started as a smaller gene therapy company called Genetics and changed their name, I guess, over 10 years ago now. to Bluebird Bio. And this is really to represent for those skiers in the audience, kind of that perfect powder day with blue skies. And this idea we're trying to kind of create for everyone. I've been at Bluebird for 4 years now, almost 5. I started at the birth of my daughter. She'll be 5 in April. And we've grown to about 1,000 employees. I started, we were about 250. So really exciting times for the company. We've grown in Europe, and we've got investigational therapies across a host of severe genetic diseases, from cerebral ALD, which we'll talk a little bit about today, but also sickle cell disease, transfusion-dependent thalassemia, and we have an oncology kind of side of the business as well, with a product in multiple myeloma, partnered with BMS. And again, none of these products have been approved by the FDA. So before I dive into some of a little bit of the details about our modality, as Ben said, there's a whole host of modalities, a very exciting time for the community. Talk a little bit about the context in which we're kind of developing our gene therapy approach. And we heard earlier today, right, a lot of great background and introduction on allogeneic stem cell transplants, and we'll learn more today about gene therapy in the afternoon. But it's really based on, allogeneic transplant, which was, has been in use for treatment of, cancer and some severe genetic diseases for 40 plus years now. And the concept here is you can replace a person's own stem cells or disease cells with those from a healthy donor, in an effort to treat that underlying disease by getting healthy donor cells. And really, with the advent of more sophisticated molecular biology, learnings in viral vector development and biology, we've really become quite sophisticated in our ability to manipulate the genome. And there's a whole host of things that you can do now. And we utilize one of these approaches, but just to set a broad context, right? There's approaches called gene addition, where you can take advantage of viral vectors. Mechanism to insert genes into the genome. In some cases, we'll talk about what we do is insert a functional copy of a gene that may be dysfunctional in disease. And there's a whole host of other, and growing every day, other kind of genetic modification toolbox, right? So gene editing, where you can make more specific edits to the genome, even getting down to the level of manipulating at the very specific gene regulation and different elements of a gene to control potentially dysfunctional or create functional genes. And this is, in our case, we're working with a cell-based therapy ex vivo, which means we take cells, and I'm going to talk a little bit about this in just a minute. We take cells out of the body and manipulate them, and then infuse them back into the body. But there's also been advances in gene therapy where some of these modalities are done in vivo, and I think we'll hear a bit more about that later today. So our investigational drug product candidates, like I said, we work across a number of diseases. We'll talk a little bit more about Lenail, which is our product for cerebral adrenoleukodystrophy, but also have products in development for transfusion-dependent beta thalassemia, sickle cell disease, and as I mentioned, our oncology business, and a program in multiple myeloma, utilizing CAR T cells rather than hematopoietic stem cells in that case. So our clinical development program for Lenail and cerebral ALD started a number of years ago with our ALD 102 trial. This study is actually now fully enrolled. We've treated 32 patients in this trial, and those patients, or a number of those patients, have now rolled over into a long-term follow-up study that we call long-term, our LTF 304. So in the ALD 102 study, those patients were followed for 2 years, following treatment with Lenail, and then they transition into a long-term follow-up study for additional monitoring and follow-up for an additional 13 years. We have an ongoing phase 3 study called ALD 104, which is currently recruiting patients in the US and at centers in the EU. The major difference between these two studies, in ALD 102, we were using a conditioning regimen known as busulfan and cyclophosphamide, and we excluded matched sibling donors from that study. And in ALD 104, it's now open to any patient with CALD less than 17 with these specific disease characteristics down here in the enrollment criteria. And we've also altered the conditioning regimen to get a better understanding of a variety of conditioning regimens with Lenail, and that's using busulfan and fludarabine. So to talk a little bit about, our particular process, as I mentioned, we're an ex vivo gene addition modality. And so what we do is we collect the cells from the patient, this HSC collection step, so hematopoietic stem cell collection. And we do this, we have to get them out of the bone marrow, and we do this through an effort called mobilization, which is done with a few molecules here, GCSF or plerixafor, and harvested there at apheresis machine. Those cells are then shipped to one of our central manufacturing sites, and we select a kind of specific population of hematopoietic stem cells for transduction with our lentiviral vector. And that lentiviral vector is carrying a functional ALDP gene in the hopes of correcting the disease. And we've heard a little bit about that this morning. Those cells are then cryopreserved, which means frozen down, tested for quality and safety, and shipped to our infusion sites for the hematopoietic stem cell transplant. So just prior to infusion, patients have to undergo a process called myeloablative conditioning. And in ALD 102, as I mentioned, this was done with busulfan and cyclophosphamide, and in 104, a slightly different regimen, busulfan and fludarabine. And the idea here is you need to kind of clear, I've heard Christy Duncan, who's a transplant physician, describe this as kind of sowing the seeds, right? So you've got to plow the field or the bone marrow to make space for these new cells. And that's the idea of myeloablative conditioning, to allow space for these new transduced cells to take hold in the bone marrow or in grafts upon infusion. And as we've learned a little bit today about the kind of science behind the exciting science that's happening across the community, it's known that some of these cells, when they engraft in the bone marrow, can make their way into the brain and become functional microglia. And this is how we think we can serve to stabilize the disease in cerebral ALD. And then those patients following infusion are followed for 2 years, like I said, and an additional 13 years in a long-term follow-up study. So lastly, and just to make space for the rest of the team here, we're really excited to share some of these newer resources that we've been able to partner with the community on to increase education about the disease, about newborn screening, about gene therapy. So first, and I think if any of you were at Hunter's Hope, we were able to kind of give a teaser on this newborn screening toolkit that was in production, and really excited to share that this has now been published. You can access it on our website. And this is really an educational material to help educate folks about the importance of newborn screening, which we've talked about, Elisa's going to talk about later today, and what we can do to advocate across the states and broadly to advocate for newborn screening efforts. And again, this was an educational resource developed in collaboration with this community and others. So thank you all for the contribution. It really is a beautiful piece of work, I think. In addition to that, we have a kind of a disease education website, Navigating ALD, which shares some patient stories, again, working very closely with the community, and all the better for it. And lastly, something that the company is really excited about is called Gene Home, a bit of a play on words, and a gene education or gene therapy education website to help explain some of the complexities that go into these therapies. And again, I think the team's done a really amazing job. Bluebird's home to a really passionate, smart group of people that like to explain the science, help other people understand, and work with the community for that. So just wanted to share some of these. You can go to all these various links. And thanks again to all of you for the partnership and the creation of these.
Wonderful.
Thanks, Ben.
Thank you, Joe. Thank you so much. So we're going to go straight to our next speaker, Richard Kim, who is from ImStem and has a very novel and promising approach.
Thank you very much. It's a pleasure to be here. My name is Richard Kim, and I'm a neurologist by original background, but I've been in clinical development for 20 years, and I'm the Chief Medical Officer of ImStem Biotechnology. And this is our first time presenting to all of you. So thank you to the distinguished members of the panel for inviting us. Just a little bit about who we are. We're quite new. We are incorporated in Farmington, Connecticut, and we started out as a company in June of 2012. The company was actually founded by two scientists who hold on to our proprietary technology and the intellectual property behind how we make our stem cell product. So we believe that we have the best-in-class, off-the-shelf, allogeneic pluripotent stem cell-derived technology and therapy for CNS, autoimmune, and rare orphan diseases. We had our very first IND approved for multiple sclerosis indication in March of 2020, literally as all of us were going into the pandemic. And we truly hope and believe that our second IND could be cerebral ALD in 2021. And very recently, we announced in a public press release that we are collaborating with another company in the stem cell space on COVID-19 treatment and acute respiratory distress syndrome. So that is our kind of like a corporate plug. And just because I'm mindful of a lot of patients who may be watching this Zoom video teleconference, I just wanted to explain our product a little bit in layman's terminology. So we are not gene therapy. We are a stem cell product. And what we do is we take a single allogeneic, meaning not from your own self, but from another healthy donor a long, long time ago. And we actually take a human embryonic stem cell from a healthy donor from 10 plus years ago, and then we use that as a single source to generate billions and billions of mesenchymal stem cells. And our product is given IV. And when many of you in the patient audience think about stem cells, many of you, might be thinking what you are seeing on the left half of the slide, which is the pluripotency of these, stem cells that can actually migrate and go into different places throughout the body and actually repair and become something different. So that would be tissue regeneration. And I start with that to just emphasize that is not what we do. So we are not about tissue regeneration. And in fact, we are on the right half of the slide. In fact, over the last 30 years, numerous publications and scientific data have pretty much established that these mesenchymal stem cells possess a lot of immunomodulatory as well as immunosuppressive properties. And it is through that angle that we deliver our therapy in an intravenous fashion. And just because of my original background, as well as the background of our founders, just historically and temporally, we are first in multiple sclerosis, but from then on, we are hoping to branch into cerebral ALD as our next IND indication. And then this is the last slide, that I have. And, I just wanted to begin with a little bit of a story in that about a year ago, when we were thinking about, where do we take our stem cell product beyond multiple sclerosis, given my development background, our next indication might have been actually inflammatory bowel disease, which is another big indication, that takes often 10 plus years for some of us on the industry side to develop. So we actually started to search, for something different. And even with my background, but an outdated background as a neurologist, who hasn't practiced in more than 20 years, I was just amazed and pleasantly surprised at how much similarity there was between multiple sclerosis and a demyelinating disease, such as cerebral ALD. So one thing led to another, and then we actually reached out to Dr. Troy Lund. And, we actually started collaborating with Dr. Lund in Minnesota already. And we have begun to generate some preclinical in vitro proof of concept study in ABCD1 deficient blood-brain barrier model.
Okay, Richard, 1 minute, please.
Okay, and we hope to be able to share some of our results in later years to come. And that's all I have.
Okay, wonderful. Thank you so much, Richard. So next, we are going to go to Meya, who is joining us from Switzerland and who has worked for Minoryx Therapeutics for quite a while and is going to present the progress on Min-102.
Yeah, good evening, everyone. So as Ben said, I'm based in Switzerland, so it's evening already here, and my pleasure to communicate where we are with you tonight. Can you see my screen, Ben?
Yes.
Okay, very good. So yeah, I'm Chief Medical Officer of Minoryx. And as my other speakers, I would love to be back to see you in person, hopefully next year. But let me just give you a brief update of our approach to CALD. So this is our drug candidate. It is called leriglitazone now with its new generic name. The other name is Min-102. Just to recapitulate for those who don't know us, Min-102 or leriglitazone is a selective PPAR-gamma agonist. Now, for the non-scientists among you, this is a class of receptors that are nuclear receptors. That means they do not sit on the cell surface, but at the nucleus of the cell. And these receptors, particularly PPAR-gamma, are involved in many biological processes. You could say around protection against damage and healing. So apart from sensitizing to insulin, it protects against injuries to neurons. It helps neurons to regenerate, and it is also anti-inflammatory. Now, our compound is a metabolite of a well-known drug, which is called pioglitazone, or Actos by trade name, that has been indicated, has been on the market for quite a while to treat type 2 diabetes. And actually in diabetic patients, leriglitazone, which is the so-called fourth metabolite, or M4 of pioglitazone, it is the main contributor to its efficacy. That means through the use of hundreds of thousands of patients of pioglitazone, we have quite a lot of safety information accumulated indirectly. Now, we chose leriglitazone over pioglitazone because it has a better penetration to the brain and also to the spinal cord. And you cannot just go to the pharmacy and get a prescription of pioglitazone to treat ALD, because the exposure and the levels in the central nervous system that are required for efficacy to treat ALD cannot be achieved with pioglitazone. This is where we are. We have two studies in XALD. The first one is our large study. It is called ADVANCE. And I'll update you where we are in a minute. This is now completed, but we have a second study ongoing that is recruiting boys with CALD. We've also recently completed a study in Friedreich's ataxia and wait for the data to come out. And we're actually looking actively into other CNS diseases. Now, what was ADVANCE? And I think several patients are probably among the audience here. So this was a double-blind placebo-controlled study with 2-to-1 randomization. That means as a patient, you had a 2-to-1 chance to receive active treatment with once daily dosing. We have centers across Europe and the United States, and we have now completed the double-blind part, and can actually say that the vast majority of patients now agree to continue in the long-term extension. And down below you saw the time schedule for the double-blind phase, but I'm not going into details here. These are the study sites. So you see that we have 7 sites in Europe, and then 3 study sites in the United States. We are on track with the development of the data from this study, and we anticipate that we can announce at least the top line results before the end of the year. I recall last year when we met near Waltham, you asked me, now when are you going to announce it? Is it at the next ALD Connect meeting? And I said at that time I cannot commit to that, but I said we will have the data before we all break for the Christmas and New Year days, and that is still on track. Now you may ask, if positive, what are we going to do? Yes, we are going to continue with further studies. One will be in female patients, and we are also looking at other patients with XALD. But I also wanted to alert you to our second study and tell you where that study fits in the disease evolution. As you all know, and hope you see my cursor, CALD starts usually age 2 years and above. Before that, patients are totally asymptomatic. Then gadolinium negative lesions develop in the MRI. Several patients convert to gadolinium positive or inflammatory lesions after first appearance, and only then usually clinical symptoms appear, and if undiscovered and or untreated, it leads unfortunately to death or vegetative state. In most of the patients, there's a spontaneous arrest in about 10 to 15% of patients. But when discovered early, hematopoietic stem cell transplant is the treatment of choice, and potentially bluebird's gene therapy will appear on the scene as the second option. And that will arrest the disease if successfully engrafting has occurred. And we are trying to get into this slot before stem cell transplant or gene therapy may be indicated. It's still quite massive procedures with myeloablation. And we want to see from very first appearance of MRI lesions until transplant, can we maybe avoid transplant, or can we at least delay it or keep the boys at a better state? And this is the question of the study. This is open for enrollment. It enrolls boys aged between 10 and 12 years with very early MRI lesions. This is an open label study. That means all patients receive liraglutazone on top of standard of care. So nothing will be delayed or withheld. We have sites open in Spain, Germany, and France. And recruitment is also open to international patients outside of Europe, and we will provide travel assistance as needed. The endpoint is disease progression defined by MRI and clinical criteria. We look at short-term arrest at week 24. And if disease is arrested, we extend it to 96 weeks, but also treatment may be provided beyond that. So as I said, that study is open. It has already started enrolling boys, and we look forward to seeing more patients on that study. And that is all that I have to tell you today. But since so many patients are there who may have joined our advanced study, I really wanted to thank all of you. I know it was not easy for you to commit to 2 years with the potential to receive placebo. Thank you for your commitment, cooperation, and patience. It was a long journey, and even more during the difficult days of pandemic when sites were closed down and we had to improvise and find other ways that we get you to the visits. So thank you very much.
Wonderful. Thank you, Uwe.
Mm-hmm.
Great progress. Okay, next we will go to David Moller of Poxel. And David, let me tell you, we can see your screen.
Great.
Thank you so much.
Thank you, Ben. It's great to be with you. I'm David Moller. I'm the Chief Scientific Officer at Poxel, and I'm happy to be joining ALD Connect for the third time. We are a small biotech company focused on metabolic disease. We're based in Lyon, France, but we also have affiliates in Boston and in Tokyo. And our focus is really on novel mechanisms to ameliorate metabolic pathophysiology that can lead to inflammation and subsequently to tissue degeneration and cell death. So currently we're focusing in our late stage portfolio on diseases like type 2 diabetes and fatty liver disease, but we're expanding into more orphan metabolic indications, including adrenoleukodystrophy. So here's a snapshot of our pipeline. I just want to give you a couple of additional points about the scope of what we're doing. We have a late stage molecule called imeglumine, which is intended for the therapy for type 2 diabetes that modulates mitochondrial metabolism. And that molecule has actually been filed with the new NDA in Japan, will hopefully be available to patients in the coming year or so. And then we have two mid-stage development candidates, PXL770 and PXL065. One of them, both of them are targeting defects involving metabolism, lipid metabolism, and mitochondrial metabolism as well. And you'll hear more from Jaspreet Singh later on this afternoon about using both of these molecules as tools to study in vitro and animal models of AMN and ALD. And those molecules in turn each represent a platform, an individual platform. In one case, it's AMP kinase activators, and in the other case, it's deuterium stabilized thiazolidine diones. In each of those two platforms, we have next generation molecules, and we're really focused on developing one or more of our next generation molecules for ALD and AMN. So I'll tell you a little bit more about that on the next slide, which is really a summary of some of the data that Jaspreet Singh will discuss this afternoon, and also a bit of the rationale and background for why we believe that these two platforms could be potentially applicable in patients with ALD and AMN. So on the left side, you can see a cartoon that depicts AMP-activated protein kinase. This is a cellular energy sensor that exists in every cell in the body, and it basically responds to low states of low energy to undergo activation. And when it's activated, it has important effects on a number of different pathways, including accelerating lipid metabolism, which could be beneficial in diseases where there's accumulation of long-chain fatty acids, for example. It also inhibits inflammation and has important effects to also deter cell death. And some of the data that Jaspreet will share with you this afternoon is on the lower left of this slide, depicting that in cells as well as in animal models, which isn't shown here, you can see that there's a very strong reduction in very long chain fatty acid cells derived from patients with AMN or ALD, and also an upregulation of compensatory genes like ABCD2 and ABCD3 that could fill in for the missing ABCD1 that's mutated in the disease state. On the right is some examples and rationale that underlies our deuterium stabilized thiazolidine dione platform. And I want to also mention this is a platform that came directly from a close collaboration and partnership we have with Therapeutics, which is a company that was founded and still being led by Sheila DeWitt, who many of you know, and we're still actively working with Sheila on this program. So the rationale for this does overlap with what Uwe just described for Minoryx with Min-102 or liraglutazone, in that we are trying to leverage some of the benefits of pioglitazone that have been demonstrated in preclinical models of AMN and ALD. But in our case, we're striving to dial out or reduce the PPR gamma activity, which resides in pioglitazone, but to emphasize some of the other pathways we refer to as non-genomic pathways, which can in turn have independent effects to enhance lipid metabolism, reduce inflammation, and potentially have protective effects in AMN and ALD as well. So the specific molecule that is our lead molecule, PXL065, I mentioned before, is in development for fatty liver disease, but we also have backup or second generation molecules in this class that are showing very promising activity in preclinical models for AMN and ALD. And an example of that data is on the lower right here, again with PXL065, the lead molecule in this case, and you can see that it can reduce very long chain fatty acid levels in the spinal cord, even better than pioglitazone in this particular case. So we're very excited to be in the game in terms of trying to develop one or both of these two novel platforms, ultimately for the treatment of AMN or ALD, and we hope to be able to bring a molecule into active development in the coming few months or year. So I will stop here. Thank you very much.
Wonderful, David. Thank you so much. Okay, next we will have Asif Paker of SwanBio. So this is a very exciting company that's partly based on Florian Eichler's research and has grown significantly since then.
Thank you, Ben. And hello, everyone. I'm Asif Paker. I
And by
work
the way,
at Swan.
Asif, we're a little bit, actually, we're good on time, so you don't have to rush too much.
Okay, that's good. I'm Asif Paker. I work at SwanBio. Prior to SwanBio, I was part of the Kennedy Krieger team that conducted Lorenzo's oil study in AMN men and women. Later, I led the gene therapy program for cerebral ALD at Bluebird Bio. Now, let me tell you a little bit about SwanBio. And here you go. SwanBio is SwanBio is a small biotech company based in Philadelphia. We are developing AAV gene therapy for neurological conditions, especially the ones where spinal cord is involved. And AMN is one of these. AMN is our lead program, where we're developing the AAV9 gene therapy for. As Ben introduced, Florian Eichler and Rachel Salzmans are two of our founders of the company. Now let me tell you a little bit about the gene therapy. Actually, Joe did a nice job of explaining what the ex vivo gene therapy is for cerebral ALD. That's the, let me get the pointer here, is the right side of this slide. Okay, so if you see the right side of the slide, this is what Bluebird Bio is developing for cerebral ALD. So gene therapy is designed to introduce healthy ABCD1 gene into the cells to compensate for the mutated gene. It could be given in a variety of ways. And for treatment of cerebral ALD, the lentiviruses is used to add the healthy gene in patient cells. And lentivector is used because lentivirus target immune cells. For AMN patients, it's not the immune cells, it's the brain, it's the central nervous system that's involved, and spinal cord is the target. So we are using AAV9 mediated gene addition approach because of the target tissue. And in this approach, that's on the right side of the picture, the AAV with ABCD1 will be infused in cerebrospinal fluid, where it will transduce the cells of the spinal cord, meaning it will add the healthy gene to the cells of the spinal cord. Now, question is this approach have been used before? And answer is yes. AAV is used in gene therapy for Parkinson's disease, in Huntington's disease, and in spinal cord, in the spinal muscular atrophy. In addition, and there are a lot of other examples. Jerry Mandel and Brian Casper developed AAV9 gene therapy that is used to treat children with spinal muscular atrophy that are less than 2 years of age. It is an approved medication and sold under the name of Zolgensma. And here it is. It is one-time dose only and target the motor neuron cells. AAV9 gene therapy in patient is planned to target motor neuron cells, neurons that's responsible for strength, for tone, and sensory neurons that are responsible for balance and position sensation. Now here I am showing the AAV gene therapy that can target the spinal cord and brain cells. Casey McGuire, Yi Gong, and Florian Eichler treated AMN mice with AAV9 gene therapy, and this approach delivered the healthy ABCD1 gene to the brain and spinal cord, as shown in these figures in the center of the slide and in the left upper corner. So these mice had no ABCD1s. These are AMN mice, so there should not be any red color. After treatment, you can see in the brain cells, there's an ABCD1 expression here. This is another brain cells. In a larger picture, I can show you the regions of brain where ABCD1 is expressed. And in the second column here, these are the spinal cord cells that are expressing the ABCD1. These were very promising results. These help us understand that it's not only a concept, if it can, this AAV9 can deliver the gene to the cells of our interest. Protein is not only expressed, but it's also functional. And you can see here that the very long chain fatty acids, the C26, decreased compared to the wild type after the injection of AAV9. In brief, this is the overview of our program, but we are we're in early stage, and we are planning for these studies. We're planning for 2 studies. One is a treatment study, and one is a non-interventional natural history study. The natural history study will provide information about the biomarkers, about the outcome measures that will help approval of this gene therapy. Second study is the first-in-human study, where gene therapy will be administered, and it will help us learn the safety profile of the drug and also help us decide for the best dose for AMN patients. This work is in progress. We are working on the details. We will share as soon as we know more and as soon as we know when we're starting the studies. Ben, thank you. And thank you all.
Wonderful. Thank you so much, Asif. Okay, and then last but not least, we will have Juliana from Viking Therapeutics, who is going to present their work on a small molecule primarily targeted at adrenomyeloneuropathy.
So first of all, thank you, Ben, and I wanted to thank the whole leadership on ALD Connect for inviting us from Viking to share with you what we have. My name is Juliana Oliveira. I'm the Vice President for Clinical Development at Viking Therapeutics. And what I will do today is share with you who we are and what we are doing in terms of development of this compound, the thyroid receptor agonist for XALD. So a little bit about Viking. We are a biotech, clinical stage biotech based in San Diego. I'm unfortunately not in San Diego. I'm based in New Jersey, but working remotely as everybody else in these days. And we are focused on therapeutics for metabolic and endocrine diseases. And we have now 2 main programs that are ongoing, both of them as thyroid receptor agonists that are selective to the beta subunit. The first one, VK2809, that you see here, is being developed for the treatment of NASH. And we did have phase 2A results that demonstrate reduction of the liver fat content and also lipids. And we have now ongoing a phase 2B trial to find a dose that will be selected to the phase 3 studies. Our rare disease program is the VK0214 and being developed for XALD. We have data in the preclinical stage that was generated before I actually joined Viking that I will share with you today. And we just started our clinical studies, which is very exciting for us. And I hope it will make everybody who is part of this meeting today feel hopeful and excited as well. The other pipeline programs that we have are in musculoskeletal and autometabolic diseases. We have also in type 2 diabetes. So now about VK0214 mechanism of action. How can we link this thyroid receptor agonist to XALD? How does that fit together? And to think about that, we just need to go back to the cause of the disease, genetic cause of the disease, the mutation in the ABCD1 gene that is needed for to encode the transporter that will bring the very long chain fatty acids to the peroxisome, and so they can be go through beta oxidation. The defective ABCD1 will lead to the transporter not being optimal, and that will bring to accumulation of the very long chain fatty acids in tissues, and that's the hallmark of the disease, as we all know. What happens is that there is a transporter that it's a backup, right? The ABCD2, it was mentioned in other presentations as well. Well, the ABCD2 has a thyroid response element in its gene. So it's been shown that the thyroid hormones and also agonists of thyroid hormones can increase expression of ABCD2, and that can mitigate the increase of very long chain fatty acids. So, well, considering all of that, at Viking, we decided to evaluate that compound with fibroblasts of patients with XALD. So that was the first study in vitro in which we did show that when put together with the fibroblasts of patients with XALD, and here they went up to 17 days of being exposed, there was a significant increase in expression of ABCD2 gene. And that led to evaluation. Next step would be in vivo, in animals. And the model used was the ABCD1 knockout mouse. And after using this compound in these mice for 6 weeks, 12 and 25 weeks, we see here that there was indeed a significant reduction of the very long chain fatty acids in the use. You can see that in these 3 graphs here, starting at 6 weeks, all of them statistically significant at 12 weeks still, and it was sustained for 25 weeks. So these was actually 2 different studies, one for 6 weeks and the other one for 12 and 25. So they confirmed the first results. And not only in plasma, there was a reduction of very long chain fatty acids, but this was also looked in key tissues. And when looked in key tissues, and that was done after the 25 weeks, it was seen that there was also an encouraging reduction of these very long chain fatty acids in spinal cord and in brain, and also in liver. That was exciting results, because that potentially could be then used really in AMN and forms of cerebral XALD. So in these experiments, we also looked at the expression of ABCD2 as well in these animals, data that I didn't bring here, but it was also increased. So considering that we had the preclinical data that was so encouraging, the company made the decision to start in the clinic. So this year, we started evaluation of safety and tolerability in our first in-human. That started in September now of 2020. This is a single ascending dose together with a multiple ascending dose, where we're going to look at safety, tolerability, pharmacokinetics, and considering that all will be favorable results, we'll start what we call the proof of concept, this number here of the study 102, that will include then patients with AMN. Will be our first target, will be AMN, that to be started hopefully in the next year. We are doing the planning of this study in parallel to the execution of the first in-human study. So in summary, this is a compound that is an agonist of thyroid hormone receptor. It's really selected for the beta subunit. It's a small molecule, so it's to be given orally. These results that we found in animals were really encouraging as early as 6 weeks, and were sustained for 25 weeks. So it does support the thinking of developing this compound, and that's why we moved to the clinics. We received an orphan status from the agency, sent our IND, started the study now in September, and hopefully we're going to be able to start the study in patients in 2021. So that's all I have. Thank you so much, Ben, and thanks again for participating in this panel.
Wonderful. Well, thank you so much, Juliana, and thank you to all the panelists for being here. and for working on our disease, either with drugs for the kids or for adults. I will, since we have a couple of minutes, I will ask just a couple of questions. So one question that's addressed to Joe of Bluebird Bio is when their therapy will be approved by the FDA and commercialized.
Sure, always a timely question, right? So I am excited to say, at least in Europe, that we have successfully submitted our MAA that's under review. So that's in the European setting, of course. And we're guiding at the moment to mid-21 for a BLA submission in the US.
Okay.
I can't comment on specific approval timelines. Obviously, have to work through regulators over the approval process, but the submission is planned for mid-21.
Yeah, so we can say it's essentially imminent. Yeah. Then another question, maybe Uwe from Minoryx can start answering that one. So a couple of folks, a couple of attendees asked for the remyelination angle, remyelinating properties of many of these drugs. What can we expect?
Well, we have seen that in the test tube in non-clinical experiments for MIN102. Obviously, we are looking at MRIs and other parameters in our ongoing or finished studies. So from that angle, I cannot comment whether it is the same in man, but from non-clinical studies, yes.
Okay, maybe Juliana, you can go next. Is that a mechanism of action for your asset, your drug candidate?
So what's the question about the mechanism?
Is there a remyelinating property in your drug?
Property?
Yeah.
I don't know. This was not yet evaluated if there is a remyelinization property.
Okay.
What was looked was really the biochemical part in these animals so far. That's a good idea, though, to look.
And maybe Asif, you can jump in.
Yeah, Ben, we haven't looked into that. We haven't. There's a work in progress that we learn from animal studies. That being said, we know from pathology that a lot of cells in the neurons are atrophied, but they are not complete. They're not dead. So especially in DRG, cells are atrophied, but they're not dead, as in Friedreich ataxia. So there is some hope of reversal of that. The cells come back to normal, but all that has to be studied, and then we can talk more. I can tell you more about it.
Okay, wonderful.
And maybe I could comment on that question, Ben. As Uwe was alluding to with pioglitazone, it's been published in animals that you can get some degree of protection from demyelination. I'm not sure about remyelination per se, but so we think that that's a potential for our deuterated thiazolidinedione class of molecules, and we're looking at that as well with the AMP kinase activators, but we don't have data yet. But of course, it's a lot harder to reverse damage than to sort of prevent the progression damage with any of these mechanisms.
Okay.
Hey Ben, just a quick comment. From the world of MS, there are many therapies, that are now on the market that actually have generated remyelinative data, and they are published. So the imaging technologies do exist for all my colleagues, where if you really want to look at in patients, it's MTR sequencing on MRI that you can look at. And MS patients actually, even on placebo, do remyelinate all the time. So in a bigger setting, where you have a control group, it's kind of easier to tell whether your active treatment group is doing something. But in rare diseases where, often trials are small, and you don't have a concurrent placebo group, it may be difficult. As for stem cells, we do have some in vitro data, showing that they do cross the blood-brain barrier, but we're actually targeting at the blood-brain barrier itself to stabilize it, whether it crosses or not.
Okay. Well, thank you so much. We have to sign off. But again, thanks from the bottom of my heart for these wonderful presentations.